Hello, I'm Ines Rivero-Garcia, currently a first-year PhD student at CENIC, the Spanish National Center for Cardiovascular Research. I'm going to tell you about part of my master thesis carried out at Ericsson Hammer's group at Stockholm University and Science for Life Laboratory. More specifically, I'm going to give you a glimpse of how drug repurposing can be applied to COVID-19 using a network module perspective. Traditionally, drug discovery has been centered in finding a key gene in a disease and modifying its function. However, this causes drug design to be a very long and expensive process and makes it difficult for diseases with few patients or a fast appearance, such as COVID-19, to have appropriate treatments. Moreover, focusing on a single disease gene might cause reappearance of the disease phenotype due to disease robustness. Some solutions to these problems include drug repurposing and polypharmacology. Drug repurposing accelerates and cheapens the drug design process by using already commercialized pharmaceuticals. Polypharmacology, on the other hand, may help to combat disease reappearance by simultaneously targeting more than one disease gene. With the advances in the understanding of the human interactome, disease has been progressively seen as a network emergent property in which the disruption of the gene interactions are the cause of the aberrant phenotype. Moreover, within a disease we can find modules or groups of genes that are highly interconnected and may be involved in the same pathomolecular mechanisms. With all of this in mind, we aim to apply drug repurposing to COVID-19 with a network perspective in order to identify the modules and the drug relevant for this disease. For this, we integrated the human interactome with disease gene associations and found the modules within each disease. Then, we mapped the drugs that could potentially target each of these modules. For more methodological details, I suggest you check the poster out. Our main result is the identification of 4 disease modules formed by the human proteins that were found to interact with the SARS-CoV-2 virus by Gordon et al. One of these modules contains 200 of the 332 genes and is also the module targeted by most drugs. Remarkably, there are several polypharmacological drugs that could act on COVID-19, but only fostamatinib is able to target more than one module. Similarly, in the other 176 analyzed diseases, we found that only one module is usually targeted per drug in diseases targeted by that drug. And also, even for polypharmacological drugs, it is usually the case that most of their targets are part of the same module. Additionally, drugs that target more than one disease are usually able to do so because their target gene is involved in more than one pathology. And as an additional example, we found more cases of imbalanced drug distributions across modules and a lack of drugs that target more than one of these modules in a given disease. To finish my presentation, I would like to highlight that in COVID-19 there are several polypharmacological drugs, but fostamatinib is the only one that can target more than one module. This imbalance of drug distributions across modules is also happening in other diseases and could be something important to focus on for designing more robust therapeutic strategies. Finally, I would like to thank Ericsson Hammer and his group, especially Dimitri, Miguel and Luca, for the mentoring and support with this project. I would also like to thank La Caixa Foundation and Fundación Margrit y Folke Perten for their support. And thank you very much for watching this video, and I hope to answer your questions in the Q&A section.